Ladies and gentlemen, welcome to our second event of the series Advanced in Vitro Models Beyond the Standard Simulation. Today we are going to talk about intestinal epithelium model. Hi, my name is Tommaso Sbrana and I'm the CEO of IVTech. In the last 13 years, I've been involved in the development of new tools to overcome the limitation of standard multiple plate or transwell system. At the beginning, I was mainly focused on the technical aspects related with the development and characterization of these new tools. And then I shift my attention onto the business aspect related with the technological transfer and the IP. Therefore, it has been a natural step to found IVTech in a partnership with some of my colleagues. IVTech has three main goals: developing and providing a new technology to allow to develop advanced in vitro models, standardizing the data, and increasing the correlation with the human reality. This is our history. So we started in 2014 discussing with the University of Pisa to acquire an exclusive license agreement related to the IP of our technology. In 2016, we released our first starter kit made by a peristaltic pump and one or more chambers, but I will describe you better in the next slides. And we signed a distribution agreement with our current Italian distributor, TwinHelix. In 2019, we decided to acquire the ownership of the IP, and we expand the distributors network. In 2020, our products portfolio was about 6 chambers and a peristaltic pump, and we had 3 line of projects to develop new products. So our business model is structured on 3 main pillars. The first one is the distribution of the technology through a distributor network. The second one is the development of engineering services to help our customer in the design of in vitro model. And the third one is the development also of the in vitro models, including the biological aspects, in partnership with some of our biological partners. Our mission is to build a 5D in vitro model. Starting from a 3D static model, we want to add a 4th dimension related with the dynamicity of the environment. So in our technology, we have the possibility to reproduce the blood action due to the flow of medium, which recreates the exchange of nutrient and fresh oxygen, as well as the mechanical stimulation onto the tissues. But this is not enough, because we know that the cell response to an exogenous stimulus is the result of a cross-modulation between different tissues. So it is important to have the opportunity to mimic this scenario also in the in vitro model. So the 5th dimension of our model is represented by the crosstalk of the tissues. Our technology is a very user-friendly technology. As you can see in this video, we can start from and we can place a plastic disc in order to fix in position a floating scaffold. Then we close the chamber and we clamp the system. The cells are ready to be placed in the incubator and mature. As you can see, this procedure is very similar to the standard protocols that you usually follow using a multiple plate system. The chambers are standardized in dimension, so we can use commercial scaffolds such as a sponge, hydrogel, even commercial reconstructed tissues. We can print the scaffold directly in the chamber or bioprint a tissue using a commercial device. The chambers have to be joined with a peristaltic pump in order to develop a dynamic environment. So the medium recreates the blood action, and it represents also the way to exchange the information between the tissues. So using our technology, it is possible to develop the so-called multi-organ approach. So we don't have only one tissue in dynamic condition, but we have a pathway of different tissues which communicates by an exchange of liquids, as it happens in the reality. This is really relevant if we consider to test, for instance, a drug, and we can test the principal effect of the drug onto the target organ, as well as the side effect onto the related tissue. The standardization of the chamber means that we are reproducible, we are compatible with the cell seeding area of a single well of a multiple plate system, and we are talking about the black chamber called LiveBox 1, or we can reproduce the cell seeding area of a transwell, and we are talking about the blue chamber, the LiveBox 2. It is important to be able to monitor in real time the experiment. So we can place in our fluidic circuit a three-way valve in order to sample the liquid, avoiding any damages for the tissue and maintaining online the experiment. Moreover, the chambers are transparent in Z-axis, so it is possible to monitor the tissues using an inverted microscope or an advanced optical platform. The glass disc, as in LiveBox 1 as well as the membrane in LiveBox 2, are removable supports. So at the end of the experiment, the user can recollect all the attached biological material and perform a post-processing in a plate or in a petri dish. We have a multi-products approach. Basically, we have 3 lines of products on the market. LiveBox 1 Suite is a family of bioreactors developed for attached cell culture condition. LiveBox 2 has been designed to mimic physiological or pathological barriers. We have a porous and removable membrane which divide the chamber in a apical side and a basal side. LiveFLOW is the name of our peristaltic pump, which is very compact and compatible with the incubator environment. In LiveBox 1 family, we have 3 different chambers, each one compatible with the cell seeding area of a different slot of a multi-well plate system. So we have compatibility with 6 well plates, 12 well plate, and 24 well plate. In case of LiveBox 2, the compatibility is versus the transwell. So we are compatible with the cell seeding area of a transwell of a 6, 12, and 24 well plate. In the last case, we are talking about standalone insert that we can place directly inside of our chambers. LiveFLOW has 2 different versions. We have the constant mode pump. The user set up a flow rate and a direction, and the pump will provide this information as a constant in a constant modality. The programmable version is a new release, and in this case, the user can decide the time span of the experiment and characterize 3 or more different periods in flow rate and direction. The pump will autonomously change these parameters, avoiding the need of the user in the lab. So due to this technology, it is possible to avoid the awful visiting to the lab in the weekend only to change or to adjust the working parameters. These are an example of GUI that will be released together with the pump. Here we have some pictures about our technology. Once the cells have been seeded in the chambers or the biopsy have been placed in our system, we can put everything in the incubator environment. As you can see, the pumper is a compact product. We have a removable drawer where you can place all the fluidic circuit. And we have just to turn on the power and switch on the motor in order to start our experiment. Let's see some application of our products. We have to choose the configuration. So In LiveBox 1, we have just one inlet and one outlet, so the liquid can flow in only one direction. In this slide, I summarize some of the main tissues that have been developed using this kind of product. In LiveBox 2, we have different configurations, so we can work in air liquid or liquid-liquid interface. We can work with the same medium on the top side and on the bottom side, or two different media, one for the apical circuit, one for the basal circuit. And we can also work in perfusion modality. So we can have a perfusion from the top side to the downside of the chamber. And this is the suitable configuration for bone model, where we are going to perfuse a very thick and porous scaffold placed onto the membrane level. Reversing this configuration, it is possible to maintain a floating condition a spheroid or a cell aggregate located in the bottom chamber of LiveBox 2. In this case, the membrane will represent a filter to avoid the flow of the biological material outside of the chamber. These slides summarize the main field of application of our technology. As you can see, we have a very widespread scenario. We start to talk about metabolism, permeability studies, as well as the test of the new material. And we have also an example of a disease model. In this case, it is relevant the multi-organ approach. So I will focus my attention in the next slides on this aspect. In a multi-organ approach, we have to design, as in a Lego strategy approach, the crosstalk between the tissues that we want to mimic. So we have to choose how to join our chambers. So in these slides, I summarize two different examples. In the first case, we have three chambers connected in series. We have a hair-liquid interface module, which could be, for instance, a skin or a lung model, and other two chambers. For instance, they could represent the vessel and the heart or the liver. In this case, we have the same medium which perfuse all the chambers. So the cells have to survive to the same medium. On the other side, we have a liquid-liquid interface. It could be, for instance, the intestinal epithelium, because we are talking about that. And we could have two different media, one for the top side and one for the bottom side. As you can see, we are able to join chambers in the apical circuit. It could be, for instance, a stomach. And in the bottom circuit, it could be, for instance, the liver. So the top circuit will represent the lumen of our intestine, and the blue part will represent the blood fraction, and where our substances will be absorbed and diffused up to the liver. This is important once we use circulating materials such as nanoparticles, nutraceutical compound, or even circulating cells as lymphocytes. The environment can be characterized also from a fluid dynamic point of view. So we can have the same flow rate for all the chambers in the first case, but we can also develop different flow rate, one for the apical side, one for the basal side, due to the design of our LiveFLOW. So the question is how to choose the suitable flow rate in order to mimic the mechanical stimulation of that particular tissue. And to answer to this question, we can develop, helping our customer, we can develop a CFD model in order to study the relationship between the flow rate and the mechanical stimulation. Is it enough to be predictive of the human reality? Well, the answer is it's an overcoming of the limitation of standard multiple plate, but it's not enough to be predictive of the human reality. We know, for instance, that tissues have certain ratios that we have to maintain, also from a dimension point of view. So in this slide, I summarized two different examples of a multi-organ approach. So let's say that we want to mimic two tissues in communication, and we can mimic this situation using one module for each tissue, the first case. In the second case, we decide to work with a free module which replicates the blue tissue and only one module for the hair liquid interface. So which is the best configuration? The answer can be found developing a mathematical approach called allometric scaling, and we can help our customer in this also in this part. This allometric scaling will scale down the dimension of the tissues from the real environment to the in vitro scenario, maintaining also the ratio between each tissues. So the cell answer will be predictive of the reality. Finally, we have to decide how to monitor our multi-organ model. So we have to decide where we are going to sample the liquid, for instance, so where we are going to join a freeway valve, how much liquid we are going to sample in order to avoid, for instance, a shift of the steady state. And we have also to decide how to monitor the tissues, considering, for instance, the transparency of the chamber. So in case of a transparent scaffold, we could be interested in a bright field modality. But in case of an opaque scaffold, we have to decide at the beginning to use, for instance, a fluorescent modality and avoiding the autofluorescence of the scaffold. Considering that this event is focused on the intestinal epithelium model, I will dedicate the last slide of this presentation to the intestinal epithelium. So here we have the tools that we would like to suggest in order to use to develop this kind of model. So the intestinal epithelium is a barrier. We are going to work with the LiveFLOW, and we suggest the use of a transparent membrane because of the opportunity to monitor the tissues in real time using an inverted microscope. The membranes are generally made in PC, so polycarbonate or PET, and in general, our customers decide to use 0.45 microns in pore size. Once we have set up the chamber, we have to join with a peristaltic pump. We suggest the use of LiveFLOW PRO, considering that during the day, we would be interested in the mimicking of the ingestion of food. So in this case, the flow rate in the lumen of the intestinal epithelium or the intestinal model will be increased. So the pump will increase this flow rate based on the program decided by the user. But this is not enough, because once we ingest food, the pressure in the blood compartment will increase. So we suggest the use of a new tools, LivePA. LivePA is a useful add-on that can be joined on the fluidic circuit, and it's a pressure modulator. So we can increase the pressure in our chamber using this kind of device. Basically, it is a piston which has on the outlet tube of the chamber, reducing the lumen of the tube. Because of this action, we will obtain an increase of pressure in one side of the light box too. So I conclude my presentation with three different papers. In the first case, the researcher decided to use our technology in order to develop a permeability study of curcumin and its effect onto the cells of the intestinal epithelium. Then, in the second paper, other researchers decided to study the effect of methylglyoxal intakes. It's a substance related with the bread and coffee onto in the digestive phase. So they mimic the crosstalk between the stomach and the intestinal epithelium. And finally, an example of metabolism, of the study of metabolism. Here, the researchers study the crosstalk between 3 different tissues, vessel, liver, and adipocytes, in order to evaluate the effect of obesity onto the inflammatory event related with the vessels. So that's all for my presentation. Thank you for your attention. I'm available for your question. And in the next presentation, we will see 2 different case of study. Thank you again.
So there are questions?
Tommaso, can I ask a question?
Of course.
Yeah, my name is Giuseppe. I just a very broad question. I see that the combination of your chambers, in principle, can push can make the system complex as you like. So I envisage that you can mimic your systems as complex as you want in the future. And I will ask about the complexity of and the link with your mathematical and fluid dynamics model. This is not a quantitative question, only a suggestion that you born from the image of the combination from your chambers.
Yeah. Thank you for this question. Well, increasing the number of chambers that we are going to connect in the fluidic circuit, of course, it increases the complexity of the circuit. Basically, the user has to be able to monitor each chamber in order to evaluate a dose response curve for each tissue. So we have an evidence in literature that our customers are decided to work up to 4 tissues connected in series. Let's say other customers decided to have also circulating material such as lymphocytes, and in this case the complexity will increase even more. From the CFD model point of view, they are useful in order to decide a priori the parameters that we are going to use in our experiment. Of course, we already have a range of flow rates that we can suggest in order to use our products. Once we decide to have a very specific application, we suggest the development of a CFD model in order to be predictive of the reality. But this can be a step forward. So in general, our customers decide to work at the beginning with the technology, and they decide to implement the engineering services in order to refine in vitro, and then they go over. The allometric approach. The allometric approach is very useful mathematical approach. Mathematical model, it has to be compared with the reality. So the response of the mathematical model has to be compared with the usability of the system. So it's important that this approach is developed by the supplier of the technology, because we know the usability of the product. So we know if the answer from the mathematical point of view is applicable or not. In case of negative, we have to decide a different approach from the mathematical perspective. I hope to answer to your question.
Hey, thank you.
Hello to everyone. Can you hear me?
Yes.
Okay, I'm going to share my screen. Can you see the presentation?
Yes, you can go.
Okay. So, hello to everyone. My name is Giorgia Pagnotta, and I am a PhD student at University of Bologna from Department of Chemistry, Giacomo Ciamician. And first of all, I would like to thank the organizer for inviting me today on this workshop on the 3D advanced in vitro models focused on the intestinal epithelium. So let's start with my presentation that will be focused on bio-inspired smart hydrogels to reproduce 3D bioprinted intestinal barrier. So a general overview about this project, and that it's a project focused on the fabrication of this intestinal barrier useful for drug screening. But at the beginning, we start with the design and fabrication of BIOINK for 3D printing techniques and application. After, we are going to move to the rheological characterization. That's a very important and massive part of this project, since when you work with the 3D bioprinting technique and with BIOINKS, it's really important to characterize and respect some kind of parameters such as viscoelasticity, printability, and also shape fidelity. And at the end, we were talking about very quickly on the fabrication of these models and the further application on drug screening. So let's start with what's the bioprinting process and technique. So as you probably know, 3D bioprinting technologies gain so much attention during the last decades. And since it's an automatic layer by layer deposition techniques that allow to fabricate 3D printed structures with high resolution in the 3 axes and with high throughput manner. In fact, at the end of the process, you can obtain these models that can also reproduce human tissues and organ if we are talking about of 3D bioprinting. What we can do with these tissues and organ at the end? They can be used in tissue regeneration field or in tissue engineering application, or as in my case, for 3D in vitro cell culture. So the main bioprinting technologies are divided, as I show in the slide, in inkjet bioprinter, extrusion-based bioprinter, and laser-assisted bioprinter. The first one is exploit the use of thermal or piezoelectric input to trigger the extrusion of bubble or droplets. The second one, that is the extrusion-based, is divided in 3 main giant categories. That is the pneumatic, the pistons, and the screw one. And the last one is probably the most expensive and also the high, that one that gives you the scaffold with high resolution is the laser-assisted one, and use a laser pulse to produce 3D droplets. So in my case, we're going to talk about 3D printing process to obtain 3D system for in vitro cell cultures. So, and the bioprinting process I'm going to use is usually the extrusion one, and exploit the use of hydrogels in combination with cells that has also already known as BIOINK. So a BIOINK is composed from an hydrogel that can be synthetic polymers or based by natural polymers, or also a blend between synthetic one and naturals. Cells can, of course, be depending on the topic and on the focus you would like to reproduce. And at the end, BIOINKS should be biocompatible, biomimetic, and should be able to support cell growth. And of course, it must be printable. At the end of the process, you obtain this 3D model that should reproduce the tissue, the unit of the niche you would like to reproduce. And in order to obtain the specific and the desired range of stiffness, you can also cross-link at the end of the process your scaffold with different cross-linking processes such as ionical, thermal, or photoinduced. These processes, of course, depend on the type of hydrogels you use, of course, and on the cells you would like to employ, since this chemical cross-linking process should preserve the cell viability. So at the end, the 3D printing process involves a lot of elements to take into account, and the most important is the rheology and also the shape fidelity of your construct at the end of the printing process, the cross-linking process that should give you and provide to you your desired mechanical properties, since, as you already know, probably the hydrogels and the artificial matrix should replace and play the same roles of the extracellular matrix. So the force and the mechanical force that the extracellular matrix apply onto the cells can, in a sort of way, affect the cell pathway. So the mechanics, it's a very key role in this process. And of course, we have to keep in mind that this system has to support cell growth. So let's start with the design of our BIOINK and what's our approach. We considered for our topic, that is the intestinal epithelium, 3 different kind of hydrogels. Collagen from type 1, equintendon, and this work is in cooperation with type 1 biomaterials that provide to us our collagen, and the gelatin methacrylate, or as known as GELMA, produced by myself in my lab at University of Bologna, and a blend of both collagen and gelatin. Why these 3 hydrogels? First of all, they are naturals, and in particular, collagen is ACM-like structure, so it's the most component of our tissues. They are biocompatible and they are able to support cell growth. And the blend, why the blend of both hydrogels? Since, as you probably know, blending polymers gives you the possibility to exploit both chemical and mechanical properties of the native hydrogel. So collagen usually is very tricky to modulate in terms of mechanical properties. Otherwise, GELMA, thanks to the methacrylate groups, is very tunable and gives you a lot of range in terms of stiffness. So the blend at the end can maintain the collagen, the chemical properties of collagen, but the mechanical properties of GELMA. How to produce a blend, or better, it's not a blend, but an interpenetrating polymer network. So by combining collagen and GELMA, at the beginning, we can, by using UV light as trigger, produce the cross-linking of collagens. So literally, we produce chemical bond between collagen chain, and after, by mixing photoinitiator, the IRGACure 2959, and again by using UV light, we can selectively cross-linking just the methacrylate group of GELMA. So at the end, this polymer will be in intimate contact, providing the suitable properties for 3D bioprinting application. So now let's move on the understanding of hydrogel viscoelasticity, printability, and also shape fidelity. So all the rheological characterization. How to make a rationally rheological evaluation of a BIOINK. First of all, we have to think of what happened of a BIOINK during a printing process. So before bioprinting process, it's inside a syringe, so it's in static condition, does not flow, but it has to support cell, of course, and it has to be suitable viscosity, because after a while it undergoes an extrusion process. So it produces a filament that should be continuous during time and should avoid, of course, clogging of the needle, and should display shortening behavior. At the end of the printing process, your 3D model should have display fast mechanical recovery, shape fidelity, and also fast gelation. So let's focus on what happened before printing process by evaluating storage and loss moduli. As you can see here, we evaluated the three blend, the three hydrogen, and as you can see here, the crossover between the G1 and G2 modulus gives you one information. The first information that there are physical entanglement that provides to you the suitable printable properties for our gel. And as you can see, collagen has the higher mechanical properties. Let's move on. What happened during the printing process? Well, one of the most important that give you information during the rheological characterization is the evaluation of the viscosity at different shear rates. What is the shear rate? Well, during the extrusion, the gel from a static condition passed through a higher shear rate condition. So all the macromolecules inside the bioinks should be aligned during the direction of the printing. So with the rheology, you can simulate this kind of process. And as you can see from the slide, with the higher shear rate, the lower is the viscosity. So this kind of hydrogel have a shear thinning behaviors and also good printability. As you can see, in these cases, Gelma display the higher viscosity at lower shear rate. So now it's also possible to evaluate the printing process, the entire printing process. So what happened before, during, and after. Since, as I mentioned a few slides ago, it's very important that your scaffold should display a fast recovery in terms of mechanical properties. So before printing process, as you can see, all the scaffold, all the bioinks in static condition displays around 1,000 to 10,000 Pascal per second, hence viscosity. After, during the printing, where the shear rate is very high, around 300 second last one, they are in a very low viscosity condition. But after very few seconds, because this process during like around 6 or 10 seconds, all the bioinks returns around in the native condition. But as you can see, the combination of collagen and Gelma, so the blend, the green one, displays the best recovery, since the Gelma not displays a very high recovery, but in combination with collagen, yes. So we can modulate the mechanical properties as you can see here. Here I report just a very example of extrusion by considering by including the fluorophore inside the bioink. And let's move on. What happened when you print something and after you have to cross-link it in order to make everything stable inside the incubator at 37 degrees. So as you can see, before the printing process, by increasing the temperatures, all the scaffold have not the best properties ever. I mean, as you can see, the Gelma does not maintain the good properties. Otherwise, collagen is stable all around the range of temperatures. The blend, as you can see, because of the presence of uncrossed-linked Gelma, start to decrease in terms of mechanical properties around 20 degrees. So it's not suitable for our purpose. So by using UV light and photoinitiator, it's possible to have a sort of stable condition during also at 37 degrees for all our hydrogel. So let's move on a bit in detail of how we produce the 3D intestinal barriers. So just a bit disclaimer about the fact that this project is very new and is also in cooperation with our biologists at the University of Bologna. So I want just to give you an overview of what we would like to do what we are doing now, and we are waiting for the first results. So our goal is to reproduce the first layers of the intestinal barrier, in particular the small intestinal barrier. and in health condition. So don't, not like tumor cells line, just in native condition, in order to have a clear idea of what happened inside a 3D model for unhealthy tissue. So the first layer is the epithelial cells. So the villi are composed of most, for 100% of epithelial cells. The inner layer is the lamina propria that is rich in collagen and fibroblast. The submucosa is composed for connective tissue and is rich in glands and macrophages. And the last part, the inner one, is rich in myocytes. So our aim is, that are reproduced in this schematic representation, is to reproduce the first layer by using BIOINK of collagen and epithelial cells. We're going to use the healthy epithelial cells. After the lamina propria, that is a very thin layers of collagen and fibroblast. And at the end, in the inner layer, we are going to use our blend, our EPN, since it's the most stiffer layer with respect to the other two, and we're going to use also fibroblasts. So we are going to increase the stiffness in z-axis and also the presence of oxygen and nutrients in the same way, but in the opposite direction. How to perform drug screening and how to simulate the intestinal dynamic flux. So at the beginning, we start to make everything inside normal multi-well plate or transwell. But when I hear for the first time, I met also for the first time Tommaso and Daniele during a workshop, I guess 2 hours ago, and we talked about the system. We thought that was clearly good and very useful to use their system, since, as Tommaso explained, and it's not my aim now to explain better his technology. We're going to use the LiveBox 2 bioreactors that is composed of 2 chambers, the apical one and the basal one, and they are divided by a porous adhesive membrane. So I just put a photo of how I would like to use and the setup of the bioreactors in combination with the peristaltic pump. So in my cases, I have to evaluate the drug absorption of folic acid at the end of all this project. So we are going to use the liquid in tangential flow, so represented in this part of the slide. And so the tangential flow in the apical part and also in the basal part, but just by changing the pressures. I explained to you here better. So as you know, the intestinal epithelium has 2 different flow. So the apical one is the lumen and the inner one is blood. So in transwell condition, you can also make the same thing, but of course, they're not in dynamic condition. So what we would like to do is to directly print inside the porous membrane by covering completely the area of the membrane in order to avoid during the dynamic condition, for example, for instance, the mixing between the 2 chambers of the flows, and using in the apical side the PB solution or aqueous solution or the media of for the cell cultures, and in the basal one the human plasma. Furthermore, increasing the pressure inside the apical chambers and decrease the pressure in the basal one in order to study and evaluate the absorption of the drug that pass through the scaffold by a passive absorption and not triggered by, I don't know, perfusion flows or some external forces. So just evaluate how the drugs will be absorbed because the tight junction presence or because the passive absorption. So in particular, as Tommaso mentioned before, I'm going to use the 0.41 micrometers membrane. And everything is possible. You have the possibility to put everything inside an incubator. So I conclude my presentation. I would like to thank my group, of course, and Professor Claudio Borghi and Cicero, the other supervisor from the Department of Medical and Surgical Science, Professor Enzo Spisny from the Department of Biological, Geological and Environmental Science, and also the group, and Dr. Luca Salvatore and Type 1 for provide to us our materials. Thanks for your attention. If you have any questions or curiosity, please ask. And thank you again.
In the chat, Giorgia, what about the next steps in your work and the challenges you plan to deal? Thanks
for the questions. Okay, now next week, in particular, we start to evaluate, to print the first layers. So we are attending and we are waiting for the histological results. And next step is to include in the inner layer the macrophages also, and try to study the interaction with macrophages and cells, since we would like to evaluate in futures what happened in healthy tissues and inflammatory disease, because as you know, probably during the inflammation, macrophages closely interact with the mucosa. So we would like to have a comparison between the two models. So up to now, we are evaluating the model, and we are trying to assess it inside also the bioreactors, because we have also to evaluate how the mechanical stability of the gel is inside dynamic condition, because of course nobody probably has problems when you have to use multi-well and to put it inside an incubators in static condition. But of course, imagine when you have to flow something from the apical side and the basal one, and of course your gel could be as possible as stiffness and region and everything, but could happen that something can be as you don't plan. So we are trying to evaluate this, and the next step is to use folic acid inside the LiveBox 2 and make a time point evaluation of the absorption, and also at the same time evaluate the metabolic activity of the cells before and after the drug absorption.
Okay, thank you, Giorgia.
Thank you.
Other questions? No? Okay, so we can go with the last presentation.
Thank you.
I would like to introduce Giuseppe Procino, professor from University of Bari.
Hello, thank you. Can you hear me?
Yes.
Okay, I'm going to share my presentation. Can you see the presentation?
Yes. Yes.
Okay, perfect. Good morning, everyone. I would like to thank first of all the Ivy Tech and the Twin Helix for promoting this webinar, putting together these interesting talks, and for inviting us to bring our contribution in the field of differentiation of polarized epithelial cells in vitro. And now I would like to start from the research project that was such a kind of starting point for our collaboration with Ivy Tech. This project is entitled BIOINKS, Establishment of a Human Intestinal and Salivary Microbiome Biobank for the dysbiosis to symbiosis. And I'd like to acknowledge all the people of the group. Besides me, there is Professor Andrea Gervino, Roberta De Zio, and Francesca Piccapane. We are all a group of physiologists at the University of Bari, at the Department of Bioscience in Bari. And a few words on the concept of polarized epithelium. An epithelium is in general a layer of cells that separates two environments, which could be, as in the case of the gut, the lumen of the intestine and the blood. The secretory or absorptive function of an epithelium is due to the specific localization of transporters and ion channels at the opposite pole of the cell, namely the apical and basolateral domains of the plasma membrane, separated by the tight gel. Without a correct polarization of the epithelium and its transporters, there is a risk of obtaining a simple layer of cells that does not reproduce the function of the native tissue. Here you can see the gut epithelium, and there are some transport that are fundamental to ensure the proper functioning of the epithelium, like the sodium-potassium ATPase pump at the basolateral membrane, which actively pumps sodium outside the cell and generates the driving force that allows the transport of many other molecules from the apical plasma membrane, like the glucose or peptides or other ions, and generates the proper functioning of the intestinal epithelium. Now, there are several options to study the phenomena of secretion and absorption through polarized epithelial cells. Tissue biopsies can be used, which are more physiological but difficult to obtain, or otherwise we can use experimental models consisting of phenotypic cell culture. Conventional 2D culture on plastic or porous supports in static condition allows to obtain epithelial that do not faithfully reproduce the physiology of natural epithelia. These epithelia are in general very flat, poorly polarized, and may not express a number of channels, transporters, and receptors critical to the epithelial function. The application of mechanical stimuli, such as shear stress, achieved through a continuous flow of culture media from the apical and or basolateral side, known as dynamic condition, allows instead to obtain epithelia that structurally and functionally reproduce more faithfully the characteristic of the epithelia in vivo. The flow can set in motion mechanosensors, such as the central cilia expressed by many epithelial cells, and the continuous removal of waste products and the continuous supply of oxygen and nutrients, as Tommaso said before, mimicking the blood circulation promotes proper differentiation of the epithelium. In the case of the epithelial barrier of the intestine, to reproduce a good experimental model in vitro, it is necessary to consider that in addition to the correct development of the epithelial layer, it is necessary to differentiate a thick layer of mucus, the one you see here in dark red, consisting of a loose layer, a loose upper layer, which is colonized by the intestine microbiota, which is here represented by these red spots, and a firm basal layer, the one in light green, that prevents the contact of this microorganism with the epithelial cells and prevents also their possible entry into the bloodstream. The composition of the microbiota, which is considered as a real organ within the intestine, can shape the mucous barrier and form these two layers of mucus. And the mucus is important to form the complete and functional barrier in the intestine. The aim of this project is to develop a good in vitro model of the intestinal mucosa of the colon to evaluate the possible interaction and effects of polymicrobial mixtures isolated from the microbiota of healthy subjects and or patients with chronic kidney disease. And the literature indicates that colon epithelia are grown under dynamic condition, for example, placing filters on lab shaking plates, develop a thicker epithelia and a thicker mucus layer that you can see here, colored in blue violet. And here comes our collaboration with IVTech, which produces, as we already heard, a bioreactor composed of culture chambers with apical perfusion only, the LiveBox 1 for cells seated on glass slides, or double apical and basolateral perfusion, as in LiveBox 2, for cells seated on permeable supports, which separates the two compartments. The device also includes a variable speed peristaltic pump to achieve the appropriate apical or basolateral flows. We have been using both of these chambers in our study to see the difference of the different culture condition using intestine cells, as I'm going to show you. And here I showed the confocal analysis of CACO2 colon cells grown on glass slides and cultured under static conditions. And although the cells show a good reorganization of the actin cytoskeleton, a polarized expression of the tight junction here stained with the antibody against the ZO1 tight junction scaffolding protein. You can see here better the reconstruction of the staining in the XZ plan here. The cells appear to be very, very flat. Here, I show you the confocal analysis of the same cells grown on glass slides and cultured under dynamic condition in the LiveBox 1 with the tangential apical flow. And in this case, the cells show a better reorganization of the actin cytoskeleton at the cell periphery. Here you can see it better in the XD scan. And a polarized expression of the tag junction. The cells under dynamic condition in the LiveBox 1 appear to be taller in this dynamic condition. And here, I show you the best results that we have achieved. And here is the same cells grown on porous filters inside the LiveBox 2 and subjected to tangential flow from both the apical and the basolateral side. In this condition, the cells appear much larger and they begin to reorganize themselves to form structures that resembles the intestinal crypts. From the apical side, the actin is organized particularly in the cortical area, indicating the formation of a brush border. And if you look here at the scan along the XG plan, the cells appear form a much thicker columnar epithelium than the cells cultivated in the previous condition that I showed you before. Now, an electrophysiological parameter that allows to evaluate the achievement of cellular confluence, a good degree of differentiation of the epithelium and the integrity of the same epithelium is the transepithelial electrical resistance, the TER. We tried to implement the LiveBox 2 with the plate electrodes connected to a measuring instrument, which is a volt ohm meter. And the graph here on the right show that the third trend by the days of growth, with a peak around the 16th day of culture. Now, a crucial point, as I said before, in the formation of the intestinal barrier in vitro is the formation of the top mucus layer. In our experiment, we used co-culture of two cell lines, the CACIO2 and endocyte models and the HT29 as a mucus-secreting cell model to simulate the composition of colognic epithelium in vivo. When the epithelium is differentiated, as I showed before, the mucus cells secrete a layer of mucus that settles on the underlying epithelium. And to measure the thickness of the mucus layer, we used two approaches. The first one is to use a patch clamp setup. The electrode that you see here is approached to the epithelium by means of a micromanipulator. When the electrode meets the mucus layer, there is a change of the impedance. Moving forward, the electrode will encounter the cell layer, recording a further change in the impedance. And by measuring the length of the path of the electrode, which is inclined by 30 degrees, the thickness of the mucus can be calculated, and we were able to calculate that under dynamic condition, the thickness of the mucus is higher compared to static condition of cell growth. Another approach that we put together was to use the confocal microscopy and image analysis. The cells here are loaded with a fluorescent dye, calcine, that here can be easily depicted in red, while from the apical side of the cell layer with the mucus, cells were incubated with a suspension of fluorescent beads, which remains, as you can see, trapped in the upper part of the mucus layer. And the confocal scan here in the XG plan, you can see in this picture, allows to appreciate the cell grown. Here, the cell layer is 20-22 microns thick. Here is the permeable support of the Lightbox 2, which is kind of 15 micrometers thick. And here, this huge layer of mucus that we were able to measure using this approach with this fluorescence bead, which is 1,100 micrometer thicker. And this preliminary, this is all I can show you for the moment. As I told you, these are only preliminary results, but in conclusion, I can affirm that all these results are preliminary. The use of the Lightbox 2 has allowed us to make enormous progress in obtaining an intestinal epithelium with the characteristics similar to those of the epithelium in vivo. If I can say, the limitation of these devices are from our point of view at the moment, the lack of basic sensor implemented in the chamber, for example, electrode for measuring for the real-time measuring of the tear, or sensor for measuring, for example, the pH, which is a good descriptor parameter of the metabolic state of the cells. Another suggestion that I would like to give in the evolution of this technology would be to increase the throughput of the system, considering that 2-3 weeks are necessary to differentiate an epithelium, and considering that each Lightbox 2 can maturate one epithelium at a time, it becomes difficult to foresee experiment that requires many experimental conditions at the same time. A multi-well plate culture system equipped with the IVTech microfluidic system could certainly meet this experimental need. Nevertheless, the image that I show you say that we are on the right path to obtain a truly physiological epithelium to make research on them. And now, thank you for your attention, and I will be to answer any question from the audience. Thank you.
That's for your presentation. And now we have questions. We have some questions from the chat. The first question is, do you think your model is predictive of reality, and how?
Well, at least in this morning, I told you our experiments are preliminary, but at least under the structural point of view, I think it's predictive of the reality. Now we are about to start the second part, which is the part in which we have to study actually the ionic transport and water transport across this barrier that we recreated in vitro. And the answer to this question relies on the result of this experimentation. But at least seeing how the fundamental transporters, receptors, are polarized in this in vitro model, I think that we are very close to the real physiology of the gut epithelium. Yes.
Okay, thank you. I have another question. Do you think your model can be used for studies of permeability, for example, for nutraceutical substances?
Of course, yes. This is the goal. And the reason why we are trying to maturate and establish this epithelium. The goal of our research in this model is to check, to put this epithelial in contact with the microbiota isolated from, as I said before, from healthy subjects or people with chronic kidney disease, because we already know that there is some toxins produced during alteration during this biosis that can affect the permeability of the intestinal epithelium. So yes, those epithelia can be used to make functional studies on the ionic transport across these barriers.
Okay, thank you. Now we have a question for both the speakers. At what flow rate did you set the LiveFLOW?
Giorgia, you go first.
As you want, Professor Procino. Okay. For the apical side, we set around, at the beginning, because we are at the beginning of our work, 200 microliters per minute. And for the basal one, around 120 microliters per minute. We are, as Professor Procino said also, since you have to use just one scaffold per time because you have just one bioreactors, we are trying also to set what is the right velocity and rate in order to have to obtain a good replication. But for the moment, this rate that I also talked with Tommaso, of course, was good for our purpose.
And we also tried many conditions and many different flows, but also at the end we found that 100 microliters per minute from the apical and basolateral side was the optimum. And I also have to add that using the LiveBox 2, you can also perform nice culture with the air-liquid interface at the apical side. It means that after a while, after you have differentiated the epithelium from both sides, you can close the flow in the apical side and expose the epithelium as it occurs, like for skin or for many other epithelia in our body, to get in touch with air. Like in intestine, we also put a very small amount of medium in the apical flow and continued to perfuse the epithelium from the basolateral side at that flow, 100 microliters per minute. And the air-liquid interface seems to complete the full differentiation of the epithelium at the end of the differentiation protocol.
Okay, thank you. We have another question for both speakers. What kind of sensor do you think would be helpful if integrated in the IVTech fluidic system?
As I said before, at least under a physiologist point of view, I think that that would be very useful to have the transepithelial electrical resistance sensors embedded into the chamber, because that way you can follow the increase of the resistance by the time, which is a good indicator of the good maturation, confluence maturation, and establishment of a good resistance, ionic resistance of the epithelium. So that would be the sensor that I think, although we tried also to make customs electrodes that can be put like the top and bottom slides of the chamber. Unfortunately, we used that material that were not transparent, so we had to make two cultures in parallel, one with the slides and one with the electrodes in parallel with the same density of cells, and look in parallel what happened to the transepithelial resistance as the cells start to grow and maturate in the chamber. That would be our first choice, but also a sensor for the pH could be useful too.
In my case, I will be honest, I have never thought about what kind of sensor you could integrate inside it, but it's a very, I think, good observation. And I think that what Professor Procino said should be very, very useful. And also, if you want to study, for example, muscles tissue, probably, or you want to use myocytes, probably a kind of sensor that gives electrical stimulus inside the bioreactor could be useful, I think. For the case for the pH, in my cases, because we like selecting a liquid by taking from the connecting tube, so I think we are like trying to deal with it in this way, but for sure some kind of sensor, as Professor Procino said before, yes. And also if you want to, I would say, study myocytes or muscle tissues or all that kind of tissues that needs of inputs or electrical stimulus or something like that. Yes, that you can like trigger on off, of course, so with an external box or an external not pump but system that you can switch on off. I mean, yes, this could be utopia and a bit tricky, but yes, in this sense, of course.
Okay, thank you. Other questions?
If I may interrupt you, I'm really happy about this feedback because we are working exactly in this direction. So we are implementing this kind of sensors and electrodes in a live box too. So I hope that we will be able to release the first prototypes in the next few months.
Good.
Great, Tommaso.